Welcome back to my channel. This is Maluk once again, your online educator. In my previous video, we tackled what the sense of touch is. Now, we're going to discuss the sense of taste. I hope you will enjoy watching and learning new things in this video. Let's proceed with the discussion. What is the sense of taste? Our sense of taste is derived from the interaction of stimuli, such as food, with the sensory receptors in our mouth and throat. Within the mouth, the tongue, and the palate, the roof of the mouth, are responsible for tasting most of that sumptuous pepperoni pizza you ordered. Did you ever wonder why your favorite foods taste so good? Well, you can thank your taste buds for letting you appreciate the saltiness of pretzels and the sweetness of ice cream. Taste buds are sensory organs that are found on your tongue and allow you to experience tastes that are sweet, salty, sour, savory, and bitter. How exactly do your taste buds work? Well, stick out your tongue and look in the mirror. See all those bumps? If you closely observe your tongue, you will see that it is not smooth, but rather rough and bumpy. These tiny protuberances on your tongue are called papillae, which is where your taste buds reside. Many of them, including circumvallate papillae and fungiform papillae, contain taste buds. When we eat, chemicals from food enter the papillae and reach the taste buds. These chemicals, or taste tanks, stimulate specialized gustatory cells inside the taste buds, activating nervous receptors. The receptors send signals to fibers of the facial, glossopharyngeal, and vagus nerves. Those nerves carry the signals to the medulla oblongata, which relays them to the thalamus and cerebral cortex of the brain. Our taste buds can contain as little as a single taste receptor or as many as 50 to 150 receptor cells. Taste buds have very sensitive microscopic hairs called microvilli. The average person has about 10,000 taste buds and they're replaced every 2 weeks or so. But as a person ages, some of those taste cells don't get replaced. An older person may only have 5,000 working taste buds. That's why certain foods may taste stronger to you than they do to adults. Smoking also can reduce the number of taste buds a person has. But before you give taste buds all the credit for your favorite flavors, it's important to thank your nose. They also send messages to the brain. Other factors help build the perception of taste in the brain. For example, the smell of the food greatly affects how the brain perceives the taste, according to the journal Flavor. Smells are sent to the mouth in a process called olfactory referral. Olfactory receptors inside the uppermost part of the nose contain special cells that help you smell. This is why someone with a stuffy nose may have trouble tasting food properly. Texture, translated by the sense of touch, also contributes to taste. Taste perception or gustation is the sensory detection of food on the tongue. Taste is the sensation that occurs in the mouth when a substance reacts chemically with taste receptor cells located on taste buds or papillae. Taste determines flavors of foods. Humans have taste receptors on taste buds in the upper surface of the tongue and the epiglottis. Taste stimuli are typically released when food is chewed, dissolved into saliva and pre-digested by oral enzymes, such as amylase, lipase, and proteases. Humans, and possibly many other omnivores, perceive nutrients and toxins qualitatively as sweet, salty, sour, savory, and bitter tasting. Simple carbohydrates are experienced as sweet, the amino acids glutamate, aspartate, and selected ribonucleic acids are experienced as savory, or umami. Sodium salts, and salts of a few other cations, are experienced as salty, acids are experienced as sour, and many toxic compounds are experienced as bitter. The set of compounds that elicits bitter taste is by far the largest and most structurally diverse, and, consequently, humans possess about 25 functional bitter taste receptor genes, T2RS. In addition, a variety of other nutrient taste qualities have been suggested, including specific taste percepts from water, starch, maltodextrins, calcium, and fatty acids. There is, however, presently little agreement on how humans perceive these chemicals and, consequently, on whether we would describe our oral experiences with them as unique tastes. Humans taste with the edges and dorsal surface of the tongue, soft palate, the roof of the mouth toward the back of the oral cavity, and pharynx. These tissues comprise the gustatory epithelia. We do not taste with our lips, the underside of our tongue, our hard palate, behind our upper incisors, or the inside of our cheeks, although young children may have taste buds in more areas of the oral cavity than do adults. The sensory organ within these epithelia is the taste bud, a microscopic rosette-shaped cluster of approximately 80–100 receptor cells, in which chemicals are detected by transmembrane receptors. The human taste receptors have not all been confirmed in vivo except for selected toxin/bitter and a glutamate/umami receptor. Nevertheless, for many stimuli there are strong hypotheses of the identity of human taste receptors based on mouse and fly research. The principal receptor hypothesized to transduce human sweet stimuli is the G-protein coupled receptor, or T1R2/T1R3, for umami stimuli it is T1R1/T1R3, although metabotropic glutamate 1 or MGLUR1, the type 4 metabotropic glutamate receptor, MGLUR4, and N-methyldiaspartate, NMDA, receptors have been implicated, and for bitter taste stimuli it is the family of putative mammalian taste receptors, T2RS. For salty stimuli there is growing evidence that the epithelial sodium channel, INAC, in part, transduces a salty taste, and for sour taste stimuli acid-sensing ion channels, ASICs, and possibly other proton detectors are involved. Whereas it was once hypothesized that these receptors should be expressed in particular zones according to presumed taste quality regions of the mouth, we now believe that the receptor expression zones are heavily overlapping in most regions of the mouth. The human tongue contains three types of taste papillae. Valate and foliate papillae reside on the middle and sides of the posterior one-third of the tongue, respectively, and contain hundreds of taste buds collectively. Circumvallate papillae comprise an arc of small ring-like structures, tiny towers surrounded by moats, in posterior tongue. Foliate papillae are slits, leaves, in the side of posterior tongue and can appear like gills in the tongue. Fungiform papillae look like small bumps or mushrooms and are scattered in the anterior two-thirds of the tongue, each harboring 0–15 taste buds. Taste buds are also located in the soft palate, non-bony palate in front of the uvula, and pharynx, back of the throat, but are in the flat epithelium, rather than in papillae in these locations. The first inset depicts the microscopic taste buds residing within the epithelium, outer layer, of a fungiform papilla. The small structures surrounding the fungiform papilla are called filiform papillae, which do not contain taste buds, and serve to make the surface of the tongue rough and help detect food textures. The second inset depicts a single rosette-shaped taste bud from within this fungiform papilla that contains dozens of taste receptor cells and contacts taste stimuli within the oral cavity via a small epithelial hole called a taste pore. The taste bud serves as the first stage of gustatory signal processing, and there are many ways in which cells within a bud communicate with one another, including electric coupling via gap junctions and cell-to-cell chemical communication via glutamate, serotonin, and ATP, among other possible transmitters. Taste receptor cells within the buds are electrically active epithelial cells that can depolarize and release neurotransmitters. Whereas these taste receptor cells are not neurons themselves, they do communicate with nearby neurons via synaptic transmission and intercellular communication using adenosine 5-triphosphate, ATP, and other neurochemicals. Taste receptor cells are continuously replaced in the bud every 9 to 15 days, to compensate for mechanical, thermal, or toxin-induced damage to the gustatory epithelia. Moreover, the entire taste bud and the taste organ, the gustatory epithelium, can be removed or destroyed and will fully regenerate, making one of the very few organs in humans capable of total regeneration. The taste system is also highly resistant to senescence and damage. It is arguably the most durable and well-defended of all of our sensory systems, as indicated by the observation that humans who truly have no taste are exceedingly rare. From the oral cavity, taste signals are transmitted to the brainstem along cranial nerve 7, facial, 9, glossopharyngeal, and X, vagus, where there is a topographical representation of the oral cavity within the first nuclear relay, the solitary tract nucleus, in which brainstem reflexes of acceptance and rejection are controlled. Strong sweet tastes are accepted and strong bitter tastes are rejected, even in decerebrate animals and anencephalic humans. As afferent taste signals ascend the brain from caudal to rostral, the information flow is split between the ventral forebrain and more dorsal thalamocortical regions where primary and secondary gustatory cortices, opercular, insular, orbitofrontal, give rise to conscious taste sensation. The ventral pathways are involved in autonomic and visceral functions, affective and emotional processing, and memory and learning. The dorsal pathways involve multiple secondary and tertiary cortices that process taste qualities, attention, reward, valence, multimodal sensory integration, higher cognitive functions and decision-making. Ultimately, the informational content and values of the ventral and the dorsal pathways are integrated. Now, what are our primary types of taste? Sweet, sour, salty, bitter, and savory are our primary types of tastes. Let's look at each of these types of tastes more closely. Sweet. Generally, sweetness is caused by a form of sugar or alcohol. Certain amino acids may also taste sweet. Scientists think we evolved to like sweetness because it helps us recognize energy-dense foods. Sweet foods are often high in carbohydrates, like glucose, which provide our bodies with fuel. Sour, sourness, or tartness, is the taste of acids. It's brought on by hydrogen ions. Often, spoiled or rotten foods taste sour. It's thought we evolved to taste sourness to identify these types of harmful foods. Salty, saltiness is usually caused by table salt, or sodium chloride, that's added to food. It can also be caused by mineral salts. Sodium is essential for electrolyte and fluid balance. So it's believed we can taste saltiness to make sure we get enough sodium. Bitter, bitterness is due to many different molecules. These molecules are usually found in plants. However, many plants with bitter compounds are toxic. Our ancestors evolved to taste bitterness so they could recognize and avoid poison. Not all bitterness is bad, though. We can typically tolerate bitterness at low amounts or when they're combined with other tastes. Savory, savory taste is caused by amino acids. It's commonly brought on by aspartic acid or glutamic acid. Occasionally, savory is also called umami or meaty. Some scientists think tasting savoriness or having an appetizing flavor helps increase our appetite and control protein digestion. Thank you for watching. Please support my channel by clicking the subscribe button, like, share, and if you have comments, please comment down below.